Welcome to today's Center for Evolutionary Medicine seminar. Before I introduce our speaker, I would like to note that Ed Young is coming next Tuesday, and there are very few seats left. So if you would like to join us for that special evening with him discussing his recent book, We Contain Multitudes, I Contain Multitudes, then you should sign up on the website, lickety-split. All right. So today's Center for Evolutionary Medicine speaker is Professor Jenny Tung, and we're very excited to welcome her from the Department of Evolutionary Anthropology and Biology at Duke University. She is a co-director of the 4-decade-long Amboseli Baboon project, and her pioneering work at the intersection of ecology, social behavior, and physiology has not only made huge advancements within the fields of primatology and anthropology, but also has broad, far-reaching implications within biopsychology and medicine. For these reasons, in the past 2 years, both the Kavli Foundation and the Sloan Foundation have recognized her with important fellowships, and she has published a daunting number of articles since 2007. So please join me in welcoming today's speaker, Jenny Tung.
Thank you so much for having me. I've had just such fun this morning, and I'm really looking forward to my conversations this afternoon. You have a beautiful center here, and it's a real honor to be invited. So I'm going to be talking to you today about some of our work on the intersection between evolutionary concepts of fitness and regulation of the genome and social behavior, which are my particular interests. It's fun to be talking to a Center for Evolutionary Medicine because it means that I can actually unabashedly start, despite the fact that I study non-human primates, with examples from the human literature which have been really motivational recently to the work in my lab. So this is a result that actually comes out of economics. Some of you may have seen it earlier this year. This was led by a Stanford economist, Raj Chetty. And what it shows is the relationship between lifespan and socioeconomic status. Socioeconomic status, it turns out, is one of the best predictors of lifespan in human populations. This, like much of the work that is done in humans, is just an incredibly impressive data set. So what went into this data set is actually 1.4 billion person years of tax records and Social Security records. So in case you're wondering, if you happened to pay taxes in the United States and were age 40 or above, anywhere between 1999 and 2014, you are actually part of this data set, which shows that household income, this is income at age 40, is a major predictor of lifespan post-40. So again, if you're in this data set, you can figure out where you happen to fall on this axis here and how long you're likely to live on this axis here. The point of this paper was to demonstrate the degree to which socioeconomic status inequalities translate into differences in lifespan. The difference between the bottom percentile and the top percentile of earners, both for men and for women, translates to a difference in expected lifespan post-40 of a decade or more. It's a huge effect size. Huge effect sizes seem to be the rule when we want to talk about social factors and health, disease susceptibility, and lifespan in humans. So this is a modified picture from a meta-analysis that was published now about 5-6 years ago by Holt-Lundstad and colleagues. The purpose of this meta-analysis was to synthesize many decades of studies on the relationship between social isolation and social integration on one hand, and mortality outcomes on the other. So this is actually a synthesis of just under 150 separate studies of different human populations. All you really need to know here is that the further this bar is to the right, the larger the predictive power of the variable on the left is for mortality. So what this is telling us is across 150 different studies of about a couple 100,000 people cumulatively, that the probability of mortality in any given year increases by about 50% in human beings. And this is true across both sexes and across all ages represented in this study, although it tends to be biased towards later years. That's really impressive, okay? So this ability to predict lifespan approximates or exceeds the effect sizes of smoking, Alcoholism, greater than 6 drinks a day, so binge drinking. It's not shown on here, but obesity is actually a smaller risk factor for mortality risk. So all of these sort of classical public service announcement things that tell you what not to do, well, what they should perhaps also be telling you not to do is, not to be lonely. And so just a few days before I came out here, some of you who read the New York Times may have seen this. This article just came out just a few days ago on the epidemic of loneliness and what currently is being done about this in the UK. It actually also rapidly went to the top of the most emailed and trending list, suggesting that there may also be something of an epidemic or interest in loneliness in the United States as well. Then, of course, the effects of our environment on how long we live and our health across the lifespan are not restricted to social factors in adulthood. Here I'm showing you some pictures that represent some particular special study populations that many of you are no doubt familiar with. From church records in 19th century Finland, studied by Virpi Luma and colleagues, the Dutch hunger winter, the major famine at the end of World War II in the Netherlands, the Great Chinese Famine, so severe restriction of food resources in 1959 to 1961 China, estimated to have caused anywhere from an excess of 15 million to 45 million deaths during that time. And actually a study ongoing right now led by researchers at Columbia and Princeton that's prospectively following individuals who have suffered early life adversity. What we have in common from at least these 3 studies is strong evidence from human populations, again, that various sources of both social and ecological deprivation, limitation, have long-term consequences, including elevated rates of metabolic syndrome and cardiovascular disease, and also long-term effects on, especially from the Finnish population, on reproductive fitness and survival. Okay. So these are all, I like to show these examples because they're big effect sizes and they affect us. And we tend to care about the sorts of things that affect us and want to know why these things that affect us, especially things like whether you're lonely, might translate into how long you live. I don't study humans. Actually, I have no projects focused on humans. All of my work focuses on animals like these. These are wild savanna baboons in the Amboseli Basin of Kenya. These are rhesus macaques. I study rhesus macaques primarily in captivity at Yerkes National Primate Research Center. And so if I'm interested in the questions that I started out the talk with, why am I studying these animals? Well, certainly the sorts of major disruptions to access to resources or the types of things that might predict social status or social isolation outcomes don't necessarily apply here. There's been nothing that I know of in the history of the Amboseli baboons that approximates the type of deprivation associated with the Great Leap Forward in China. Right? And none of these animals actually smoke a pack a day. So why study them? Well, actually, that difference allows us to ask whether there's something more fundamental going on, perhaps conserved across evolutionary time, in why these particular types of exposures, especially things like differences in social status or differences in social integration, have such remarkable effects on things that we care about, things that we might call health in humans, but often in these non-human primates are fitness-related traits. If, in fact, there is some level of conservation in how these animals respond to sources of adversity, then they're also useful as models for understanding the mechanisms that link those predictive factors to the outcomes that we observe. And in particular, we have some ability to pursue the types of studies that are impossible in humans in these kinds of animals. So I'm going to talk half about baboons and half about macaques. First about what we now are starting to understand about the effects of early adversity in the baboons across the life course, of the long-time effects of early deprivation. And second, some work that we are hoping will come out soon on some potential mechanisms that might link social status differences to downstream outcomes. So in the baboons, as Katie mentioned, I'm fortunate enough to be part of a long-term study that's now been ongoing for about 45 years, started in 1971 by Jean Altman and Stuart Altman. And this study focuses on a wild population of baboons in southern Kenya, right near the Kenyan and Tanzanian border. I currently co-direct the study with Jean Altman, who's at Princeton, Susan Alberts, who's also at Duke, and Beth Archie, who's at the University of Notre Dame. What this study allows us to do is ask about sources of environmental variation that we believe to be important to the baboons based on work that's been done already, and their long-term consequences for mortality rates in this population. That's because the basic study design has always been follows of individually recognized animals from their birth up until their death. So recently we decided to look at the collection of early life adverse experiences that we think might be most important to these animals. So in particular, we focused on their social status, so the place in the dominance hierarchy that a kid's mom occupied when that kid was born, whether or not a baby baboon's mother was present throughout its early development up until it reached reproductive maturation, whether or not these animals were born during a time of drought. Ambacelli is a highly variable environment, both seasonally and year to year. You guys will really appreciate this. So when I try to give you give people a sense of how dry it in fact is in Ambacelli, I say it's like basically in our dry years, it's like Tempe, right? It's basically like a desert environment. But the annual rainfall that we observe year to year can range from that to 4 times more than that. We looked at resource competition, in this case indexed by the number of adult animals in a social group around the time of a kid's birth. We looked at maternal social connectedness, so whether a kid was born to a female who had strong social partners or weaker social partners, and also whether the kid was faced with competition from another sibling when it was still at a fairly vulnerable time in its development. So let me give you a little bit better of an idea what our data set looked at. So this is a very rough timeline for baboon females, life history. We're focused particularly on females here, where we have better data on time of death. So they're born and they hit reproductive maturation, menarche, around 4 years of age. And then our adults who live up to the longest living animal in our population on record, 27 years of age. So this is their reproductive period. And unlike humans, but like other non-human primates that we know of, they don't hit menopause. They don't hit reproductive cessation that is temporally separated from age in which they're likely to have other sorts of organ systems failing. So we are looking at how many adults are in that group when the kid was born and the social status of their mother at that particular time point, but both of those tend to remain pretty stable, at least over the course of a year or so. We're looking at the cumulative rainfall across the first year of that animal's life, which in past work we found predicts fertility of females later on in life. We're looking at competing younger siblings. Okay? So in this case, we're defining that as another kid born to an animal's mom within a year and a half of that animal's own birth. So baboons have a gestation length of about 6 months. So that means that that female is conceiving within the first year of that kid's life, which means that she was likely cycling. Baboons tend to go through multiple cycles like humans before they conceive, which means that she was pushing that kid off pretty early. So this is the bottom quartile in our population. I know it differs for some of the other populations that folks study here. Social connectedness, which we measure as relative rates of grooming, the most common affiliative behavior that you observe in baboons. This is averaged for a kid's mom across the first 2 years of life, and maternal loss any time in that time up until when the kid hits reproductive maturation. Okay. And so our data set here focuses on 196 females watched across their full life courses from birth till death for 73 of them. The rest of them are censored data, great censored data. Okay, one of the things that's, I think, useful about looking at these kinds of data in Ambacelli is that, unlike in human populations, so sources of adversity in human populations tend to be correlated with one another. So that if individuals are born in a low socioeconomic circumstance, they often face other sources of adversity as well. In Ambacelli, these 6 predictors are actually not very well correlated with each other. Most of them are not correlated with each other at all. And all of the R-squared values are less than 4%. So we think that we're looking at relatively independent sources of early adversity. This is how those sources of adversity are distributed in our population. We have about 1/5 of our animals who never experienced any of those sources of adversity as kids. We think about them as our silver spoon females. And then we have about a third of them who experienced one of these things, another quarter who experienced about two, and a few animals who were really hit hard by multiple of these occurrences at once. Okay. So I'm just going to cut straight to some results here. If we model mortality risk as a function of all 6 of these predictors individually, we get a highly significant result that explains about 11% of variation in lifespan. Within that model, only 2 of our predictors are actually significant. It's these guys. So losing a mom is really, really bad for a baby baboon. Okay? Losing a mom elevates mortality risk at any given time by about 3-fold. And this is after they hit reproductive maturation. This is when they're 15, 16, many years later. Okay? And having a competing younger sibling turns out to be pretty bad for you too. Perhaps we speculate because you're competing for maternal resources. So these may have some similar kinds of effects. Okay, all 4 of the other effects we looked at actually they go in the predicted direction, right? So lower status kids who are in more competitive groups, who have more socially isolated moms, who are born in a drought, all do tend to have elevated rates of mortality. They just don't hit significance. So then we were, again, inspired by some of the work that comes out of the human literature, which has made the argument that an alternative way of looking at sources of adversity or the consequences of adversity is actually to drop the nuance of every single predictor, but actually just ask how many of these bad things happen to the animal overall. Let's just add them up. Let's tally them up and ask if that by itself has a predictive effect. So we tried that. And these are our results. And we found these results stunning. What this is showing you is age at death and the proportion of individuals in a given category. So silver spoon females in dark blue, individuals who experience one adverse event in light blue. Yellow is those two event kids. And these are the kids who got hit really hard. The proportion of individuals in that category surviving up until any given age. Okay? And so what this tells us is that the median age at death in our population for females who make it to reproductive maturity is about 18.5 years. For females who experienced 3 or more sources of adversity, their expected lifespan is just under 9 years, about a 10-year difference in lifespan. Just to give you an idea of the scale, we tend to use a rule of thumb, rough translation, about 1 year of human lifespan to about 3 years of, 1 year of human lifespan, 1 year of baboon lifespan to about 3 years of a human lifespan. So that's like 30 years difference in lifespan. This is a big enough effect size that we were kind of like, oh, we should have probably noticed this, and, without having to do analysis. But it goes to show that sometimes if you look at your data a different way, you see things that are remarkable. Okay, so one possibility is that this effect is completely driven by those 2 sources of adversity I told you were really important in our individual effects model, right? The maternal loss and competing younger sibling. So we asked, well, what if we just take those guys out? Well, we can take those out and produce a new index for cumulative adversity that goes from 0 to a maximum of 4 possible adverse events. And it turns out the relationship weakens a little bit, but actually the difference in expected lifespan between the median and those kids who got hit really hard by multiple sources of early adversity is still actually just about the same. So what this is telling us is that our cumulative adversity models, by just simply adding up their tough experiences early on, are picking up signals in those other sources of adversity that we can't pick up when we just try to model them individually. Okay, what do we think is mediating this effect? Well, the fact is we can't account for most of that huge effect. But one thing we were interested in was based on a paper that my colleague Beth Archie led just a few years ago that showed that social relationships, basically grooming rates in adulthood, also predicted mortality risk for ambiceli females. And this was true for both those females' relationships with other females and those females' relationships with the opposite sex males in their group. So we wondered whether social connectedness might mediate the effect of early adversity on lifespan. And it turns out that our cumulative risk index, again, number of adverse conditions faced early in life, does significantly predict whether a female in adulthood will be socially connected to other females, but has no effect on her social connectedness to other males. And I'll note, of course, that even in this significant relationship, there's a lot of noise around the mean. So this tells us that one possible way that early adversity may have consequences for mortality is by affecting how well females are able to establish relationships with other females as they age. But it also tells us something perhaps a little bit more hopeful, which is that, since we know that this variable, social connectedness to adult males in particular, also affects lifespan, that there may be some scope for buffering, right? So maybe females can help offset the costs of early adversity by forming strong social connections with males or with other adult females. Okay. Finally, we wanted to know what the consequences of these effects were for lifetime reproductive success. So this is the number of surviving offspring a female had across her whole life if we were able to observe her up until her death. So this excludes all our censored data. This is the age at which she died. And there's a remarkable relationship. This explains almost 90% of the variance in lifetime reproductive success. Early adversity is bad for these females. It truncates their lives, and because in Ambicelli, a female puts out another surviving offspring approximately every 2.1 years, it means that the earlier you die, the less fit you're going to be. So this has some major consequences for female fitness. So what do we think what's going on here? Well, I think there are 3 points that I take away from this. One is that this method of looking at adversity, sort of adding up the experiences that animals face, may give us some explanatory power that we might not have from using more classical approaches modeling each of them individually. It leaves some interpretive it leaves us with some interpretive questions about what's actually driving it. But what the folks who pioneered this work in humans would say is that the reason that we get more explanatory power looking at cumulative adversity is because there may be some complex interactions between different sources of adversity that we're simply not able to pick up, especially given limited sample sizes, by modeling all possible pairwise or three-way interactions between our variables. So that's a question that we're interested in continuing to assess. Second, so this actually comes from a study of adverse childhood experiences in humans that has shown prediction from premature mortality. In the human literature, the argument is that these adverse child experiences influence aspects of biological development, but ultimately are mediated by adoption of health risk behaviors, particularly access to illicit drugs and smoking behavior and alcoholism and risk-taking behavior and that sort of thing, that then translate to early death. Many of those classical health risk behaviors are excluded in the baboons. They just don't do those sorts of things. They don't smoke and they don't drink and they don't do heroin. And so it suggests some direct biological pathways, potentially, between early life adversity and mortality rate that we can't really get to by studying human populations alone. And finally, this is Ambicelli. This is a savanna environment that we think is similar to the environment in which hominins evolved. And what we're showing you, so in as much as that parallel extends, what we're showing you is data that suggests that there's limited scope for adaptation to allow these baboons to adjust to these adverse events they experience. They're not compensating very well because when we look at how early adversity translates into fitness, they're doing worse. Early adversity is bad for you. Early adversity does not seem to position you to be particularly robust later in life in the face of more adversity. And this dovetails with a paper that we published last year arguing that developmental constraints, rather than predictive adaptive responses, explain the physiological early life effects of poor quality early experiences. And so in that paper, we argued that animals who experienced early life drought just tended to do worse overall than animals who did not, regardless of the quality of their adult environment, where our outcome variable in that case were a set of fertility-related variables. Here, we're replacing fertility as a proxy for fitness with an actual direct measure of lifetime reproductive success, of as good of a measure of fitness as we can usually muster. And we're extending this drought result to encompass cumulative early adversity, lots of different sources of adversity that baboons can face. And potentially, we argued that social status might have buffering effects against drought in that earlier paper. Here, I meant to change this. We have just an inkling that we really haven't tested properly yet, that early life adversity might be able to be buffered somewhat by social relationships, social integration, when these females are adults. Okay. Oh, I did put it there. Okay, that's the replace. All right, so here I've drawn this link between social interactions and fitness outcomes, and it's hard not to slip into talking about it like this. I've talked about it, I'm sure, in a way that implies direct causal effects, social causation, right? What my colleagues in social sciences have called social causation. But there are other routes that can connect this left box with this right-hand box. You know, one is this health habit, health mediation sort of effect, and another that you guys have probably already thought about is a reverse causal effect, the possibility that animals who interact less or who are lower status might in fact be animals that are not in as good of a shape to begin with, right? Certainly when I'm feeling ill, I'm less prone to socially interact with my colleagues. And so distinguishing between these alternative pathways is crucial, especially if we want to understand human health, because it gives you an idea about where you should be intervening, right? Causal pathways are important. So when we study non-human primates, we can exclude to some degree these mediation effects of the classical health habits we talk about in humans anyway, but in Ambicelli, it's actually quite difficult for us to differentiate between direct causation and a reverse causal pathway. So that is why I study captive animals, where we have a little bit more control over their social environment and can try and get to direct causal relationships. And these are the animals I study. These are rhesus macaques, another gregarious primate that lives in social groups, forms social bonds. Here are some primates, some rhesus macaques bonding right in front of you. And we were able to show, as part of my postdoc work, that variation in one aspect of the social environment, dominance rank, social status, exhibited strong and pervasive associations with gene regulatory variation in immune cells. So here what I'm talking about is the activity of genes expressed in the peripheral blood, in lymphocytes and monocytes, really. Now, what we wanted to do when I started my lab, so the work I'm telling you about has taken 5 years, was actually do a little bit more than talk about association, but actually ask whether these effects are in fact causal. So this is the study design we used. It turns out that in captive rhesus macaques, for reasons that I do not understand, when you form new social groups, will adopt positions in a rank hierarchy that are related to their order of introduction into a social group. So in other words, if we took animals from the large breeding colonies at Yerkes and introduced them sequentially into a brand new social group, the first ones that go in will tend to be the highest ranking, the highest status animals in that group. The second one in will tend to be the beta. The third one in becomes third ranking animal, and the last ones in are the lowest ranking animal. We form groups of 5, so we have a linear hierarchy of 5 females, and then that allows us to replicate the experiment multiple times. We formed actually 9 different social groups in this experiment, so 45 different animals. This by itself, we think, does not establish causality, because this paradigm is good, but it's not perfect, and it leaves room for individual characteristics to influence the rank of these animals. So what we did was watch groups like this for a year, and then form new groups using exactly the same animals. So we took all of the alpha females and put them in a group together, and all the beta females and put them in a group together, and all the third-ranking females and put them in a group together. This is a highly hierarchical species, and what happens when you do that is they form a new rank hierarchy. In fact, they do that quite quickly. So this allows us to maximize the possible rank changes within the animals we study. We see females when they're high-ranking and also when they're low-ranking. So we constructed those first groups in 2013, went for a year, constructed these second groups in mid-2014, and then followed them again for another year. Just to give you an idea, this is the formation of one of our groups. This is a measure of dominance rank, where high is high status, low is low status. This is the timeline. Each of these lines is a female who's being introduced sequentially, and what you can see, yes, is that the hierarchies actually form very, very quickly, and then they remain stable over time. That stability is typical of rhesus macaque social groups. Okay, so what did we do with these animals? I'm going to tell you about 2 data sets in particular. So we collected behavior throughout to be able to monitor status and grooming interactions and so on. And then we collected samples from both the animals in phase 1 and again from the same animals in phase 2. We again collected blood samples, but we were really concerned about the fact that different types of blood cells express different genes anyway. That's what makes them a T cell or a B cell. They all have the same DNA. They're just expressing genes differently. So what we did to overcome the possibility of confounding gene expression effects with tissue composition effects is use flow cytometry to sort the 5 major cell populations in peripheral blood: monocytes, natural killer cells, helper T cells, cytotoxic T cells, and B cells. This allowed us to not only get over the possibility that we're really just seeing changes in cell type composition, but also to ask questions about what kinds of cell types with different types of functions in the immune system are most sensitive to the effects of social status. Some of these cell types are important in innate immunity, some of them are important in adaptive immunity, some of them are first lines of defense against virus, for example, others promote inflammatory signaling in response to bacterial signals. So we wanted to know what actually is getting hit by differences in social status. Because we sampled in both phases, we were also able to ask whether if you change your social status as an adult, your gene expression profiles change with you. How plastic are these effects over a relatively short period of time? The second thing we did focused on samples collected in phase 2, and this was to get at the question of whether these differences in gene expression that we expected to see matter in the first place. Maybe we can all walk around with different patterns of gene expression in our B cells, and when the rubber hits the road, when you're actually faced with a pathogen challenge, it matters not at all. So what we did was simulate a pathogen challenge with lipopolysaccharide, which is a component of the gram-negative bacterial cell wall. We drew blood into tubes that contained just cell culture media, or tubes that contained media spiked with lipopolysaccharide, with LPS. And then we cultured those tubes in parallel for 4 hours at body temperature in an incubator, and like basically a rhesus macaque, but incubator shaped. And then we harvested the cells from both of those samples, paired samples for each individual, to ask, okay, does differences in social status actually affect your response to infection? Okay, so again, I'm going to go straight to results. Our major form of data generation here was RNA sequencing, so generating genome-wide gene expression samples. So I'm showing you data here for 440 RNA-seq samples from that cell type-specific data set from the same 45 females in phase 1 and phase 2. And this hopefully reassures you that we sampled the cell types that we meant to sample. So the major axes of variance, as expected in the gene expression data, separate out those cell types with closely related cell types that differentiate late in hematopoietic differentiation, like helper T cells and cytotoxic T cells, very similar to one another. It does turn out that some of these cell types are more common in high-ranking females than low-ranking females, so that made us pat ourselves on the back that we actually separated the cell types, so we could really differentiate different levels of regulation of the immune system. Okay, so what about gene expression within these cell types? So these are each data for each of the corresponding cell types, and what I'm showing you here is actually the p-value distribution for testing the effect of social status on gene expression level in each cell type for about 8,000 to 9,000 expressed genes in the genome. And this black line shows you our null expectations if there is no relationship whatsoever. So what we're looking for is a peak of low p-values, right, relative to the null expectations. We don't want it to look like this, or, well, if it looks like this, then it suggests there is no signal of social status, dominance rank, on gene expression within that cell type. And so by just looking at the peakiest distributions, perhaps you can tell that we saw very, very different levels of signal, depending on the cell type we were looking at. So about 1,600 or odd genes were affected by rank in the natural killer cells, and about 300 or so genes in the helper T cells, and we saw very little signal in the other cell types. Okay, so one of the problems with going directly from that to saying, okay, so we have really cell type specific effects of dominance rank, is the fact that when controlling for multiple hypothesis testing, you can actually have really similar data. Like this is the same gene in 2 different cell types, and they look pretty similar, right? It looks like there's a negative correlation. One is highly significant and one is not, because multiple hypothesis testing and the way we did it depends on the whole distribution of evidence for that cell type. So we wanted to push our analysis a little bit further by using a meta-analysis to distinguish when things were truly cell type specific and when they were shared across cell types. We used an approach developed by Matthew Stevens and colleagues called meta-analysis, with subgroup heterogeneity. All you need to know about that is that these differences, in this case, this is a data set on genetic effects on gene expression across 3 different cell types. If you did individual analyses for each cell type, would tell you that only this one is significant when you can just eyeball it and go, well, that doesn't look different. Meta-analysis will tell you that this effect is shared across all 3 data sets. And so in our data set, what we were asking is, do we have genes that really have no effect of social status across all cell types? Or do we have really cell type specific effects only in natural killer cells? Or do we have some other configuration of the 32 possible configurations, of ones and zeros you could have here? And here's our results for all of our rank affected genes. This is the configuration. Basically, I just took those horizontal boxes and turned them vertically. And what it shows us is that still the best evidence is that differences in social status very specifically affect baseline gene expression in natural killer cells, followed by genes that are affected in both NK and helper T cells. So this is a remarkable amount of cell type specificity relative to other analyses that have looked for effects shared across tissues or not. Okay, so perhaps what's more interesting from a behavioral perspective, which is sort of the background that I come from, is what's going on here in the first place. Dominance rank, social status, that's just sort of like this proxy measure for things that affect their lives actually in day-to-day interactions. What we know is that higher ranking females, that's on the right-hand side here, are targeted less by other females for harassment. And in addition, in our system, the higher ranking a female is, the more often she engages in positive valence affiliative interactions. More often she grooms. Those 2 lines just show you the data from phase 1 and phase 2 separately. Okay, so do either of these things explain the effects that we observe? Well, so we can posit that dominance rank is affecting gene expression, possibly through its effect on agonistic behavior, or possibly through its effect on affiliative behavior. And so what we asked is, if we have a relationship for a rank-responsive gene between dominance rank and gene expression, if we take into account, for example, rates of grooming, does that effect size attenuate, suggesting that part of the original effect was because of how much they groom each other. And we can plot that this way by a highly non-zero beta to begin with that shrinks in size after we take into account that mediation effect. Similarly, we can ask about something that goes in the opposite direction that's negatively correlated and how it may be attenuated by taking into account, for example, rates of harassment. Okay? So I want to show you that simply because we didn't do this for one gene, we did this for every gene that was rank responsive in our data set, and that's what it looks like. Each of these is showing the original effect size of rank and then the mediated effect size of rank after either taking into account received harassment or grooming in helper T cells and natural killer cells, respectively. So this is a little bit hard to parse, other than maybe you can see that there's like lots more green dots here. Those are significant effects. Then there are orange or beige dots here. And what that can be summarized as the percent of rank-responsive genes that we think are at least partially mediated by these 2 effects. And I think the takeaway for us from this is that for helper T cells, approximately the same number of genes were mediated by harassment as by grooming effects, about negative interactions and positive interactions. In natural killer cells, where we see the dominant effects anyway, actually grooming was explaining way more of this pattern than harassment. And we tend to think about social status, or at least I think a lot of primatologists tend to think about social status in rhesus macaques as really about harassment. They're just kind of mean to each other. And this is suggesting that a lot of the effects from the immune system we're seeing are actually a byproduct of social status driving grooming interactions instead. Okay, so what if you go up in rank, or what if you go down in rank? Do you change? And it looks like you do. So each dot here is a gene that is significantly rank responsive, plotted by its effect size in phase 1 and its effect size in phase 2. They're both significantly positively correlated, and most of these effect sizes are highly concordant in both of these cell types. So it suggests that if you were low ranking and now you're high ranking, your gene expression profile changes in the course of weeks to months. Another way to look at that instead of gene-wise, I find this helpful, is to think about it in the space of individuals. So every dot here is an individual in our study. They're replicated for the red dots and the blue dots. And this is showing how much they increased in rank from phase 1 to phase 2 or decreased in rank from phase 1 to phase 2. And this is the change in gene expression levels for rank-responsive genes. And it makes this beautiful X. That is, females who increased in rank from phase 1 to phase 2, now they express those rank-responsive genes on average like they're supposed to as high-ranking individuals, and vice versa. So it looks like these effects, unlike perhaps effects of early adversity, are actually really, really sensitive to the current environment, at least in the immune system. Okay. So does it matter for the response to infection? So again, I'm just cutting straight to a PCA of the data. These are the data for matched control and LPS-stimulated cells from the same individuals. And what I hope is very clear immediately is that the first axis of variation, as we expect, separates out control samples from LPS-stimulated samples. It turns out your cells, when they're faced with something that looks like bacteria, really respond, as they should. What was very interesting to us is that the second axis of variation in our overall data set correlated very strongly with dominance rank. Okay? And so in both of these colors, the darker colors are high-ranking individuals and the lighter shaded ones are low-ranking individuals. Okay. So again, we can go back and ask about what this looks like on a gene-by-gene level. So this is every gene that we measured. It's the dominance rank effect in the control condition, the dominance rank effect in the LPS-stimulated condition, and it's positively correlated, again, as we would expect. But there's this interesting excess scatter of genes down here. Okay, so these are genes for which the effective dominance rank on LPS has become much greater in the the effective dominance rank on gene expression is much greater in the LPS condition than it was in the control condition. That's an interaction effect between the condition in which we measure gene expression and dominance rank. And particularly, they're all in this one direction. What that translates to is a bigger effect size of LPS for low-ranking individuals being treated with LPS relative to control than for high-ranking individuals. Low-ranking individuals are changing gene expression much more between conditions. And actually, I think you can actually see that on the PCA plot if you look back at it again. These high-ranking individuals do separate, right, from control and LPS samples, but less so than these low-ranking individuals down here. Okay, so how can those interactions occur? Well, here are gene sets that are upregulated in the LPS by LPS. Here are genes that are downregulated by LPS, and genes that are more lowly expressed in high-ranking individuals in this column, genes that are more highly expressed in high-ranking individuals in this column. It's a genome-wide data set, guys. We can find examples of all of these patterns. In fact, we can find abundant examples of all of these patterns. This is the number of genes that fall in each of those categories. So to ask about, biological sensibleness, biological coherence, we asked whether particular types of genes tended to be enriched in each of these categories. And it turns out in those bottom categories, there's nothing. There's no coherent pattern. But in both of these top categories, we find a coherent pattern that's dominated by inflammatory responses in these types of genes and type 1 interferon signaling in these types of genes. Okay, and in fact, this is a gene involved in the inflammatory response. This is a gene involved is that actually interferon regulatory factor 9. And so because it's a little complicated to keep these categories straight, I'm just going to call these low-rank genes, more highly expressed in low rank. These are high-rank genes, more highly expressed in high-ranking individuals. Okay, so I'm sorry to spring this on you towards the end of the talk, but I wanted to show you this pathway diagram. So this is the LPS signaling pathway through toll-like receptor 4. So lipopolysaccharide primarily signals through this particular receptor, which can signal through a MyD88-dependent pathway, the left-hand side, that promotes a pleural inflammatory program, or a TRP-dependent pathway that promotes an antiviral program. And lots of those genes that are more expressed in low-ranking individuals are over here. Lots of the genes that are more highly expressed in high-ranking individuals are over here. And in particular, we're interested in the fact that these two pathways end up signaling through particular transcription factors. And it turns out that if we look at transcription factor binding sites in accessible DNA, DNA that's open to protein binding, that the ones that are enriched in near genes more highly expressed in low-ranking individuals happen to be NF-kappa B, and the ones that are more highly enriched in genes near high-ranking individuals tend to be these interferon regulatory factors, as we would predict. So it really looks like low-ranking individuals and high-ranking individuals are simply using different arms of this pathway, one involved in antiviral defense and one involved in pro-inflammation, that antibacterial defense. Okay, I think I made that point, so I'm going to skip through this slide in the interest of time and just tell you that what we think we're seeing in macaques is that low-ranking individuals experience a different behavioral environment. They're harassed more and they're groomed less. They experience differences in glucocorticoid physiology that I haven't talked about today. And this stuff translates into the function of their immune system on multiple levels. both the cell types that are circulating in their bodies and the gene expression programs within those cell types, their response to infection when those cells are actually threatened by an external event, and also the specific usage of signaling pathways within the cell. So together with the baboons, what do I think we're seeing? Well, I think we are seeing good evidence that major predictors of social and environmental adversity in humans translate to other non-human primates, translate to our closest living relatives, which means we can study them to understand both the evolutionary history of those responses and use them as models for understanding the mechanisms through which we get sick because of exposure to these sources of adversity. Based on the baboons and the macaques together, we think that the ability to compensate for exposure to early adversity is limited, but maybe that's less so through exposures to certain types of adversity in adulthood, where we see a lot of plasticity in these effects. Finally, I think that our data together tell us that exposure to adversity is it's bad. It limits fitness, and it's unlikely that animals have the adaptive capacity just to completely bounce back from that, even if they're put in a similar environment in adulthood. However, where I think evolutionary explanations become very important, we've been thinking about this with macaque data, is understanding why these sorts of constraints, why these responses happen to manifest in the way they do. Some people, for example, have speculated or tried to test the idea that some forms of immune defense are more costly than others, and so that may map on to the differences between the low-ranking responses and the high-ranking responses to infection that we observe. So with that, I just want to thank the people that I collaborate with, the baboon work, the macaque work, especially Susan, Jean, and Beth, and Luis Barrero and Mark Wilson on the macaques, my lab, and funding. And sorry I ran late, so I'll take questions if there's time. Minutes. Are there any questions?
Thank you. Very interesting. You mentioned a little bit glucocorticoids. Does it appear that some of the social impacts on immune function are those being mediated by the glucocorticoids in a stress response, or does it look like there's more going on than that?
Yeah, I mean, that's a very reasonable hypothesis, especially given the long history of work on socially induced stressors and glucocorticoids. So we measured glucocorticoids in various ways, cortisol in various ways, with these animals. And what it looks like to us is that if you just take diurnal cortisol measures, we see no effects of rank, but if we do measures of glucocorticoid sensitivity, we do. And again, like the gene expression data that I showed you, when females go up in rank between phase 1 and phase 2, or down in rank between phase 1 and phase 2, their measures of glucocorticoid sensitivity actually shift exactly as we would predict. So that supports the idea that CORT might be important here, in particular because CORT does things like directly regulate NF-kappa B, that transcription factor that I talked about in the end of the talk. So we used a little bit of our glucocorticoid sensitivity data actually in revisions for this paper. I'm assuming you were not the reviewer, but I can tell you the answer to this better now this week than I could 2 weeks ago. So we asked if that mediated some of the responses to infection that we observed. And we do see significant mediation, more than expected by chance, of a glucocorticoid phenotype on the rank effects on LPS response that we observe. It doesn't affect a lot of genes, but at least it affects some that kind of make sense to us in terms of pro-inflammatory signaling. I think that, however, I just don't think our data are clean enough to say how much really glucocorticoids contribute. It's a tricky question to ask because when glucocorticoid phenotypes correlate with rank, then there's almost going to be some statistical signal of mediation. And there's a lot more that we could actually do to get at that question. Like one thing we're doing actually is doing synthetic glucocorticoid challenges of cells from low and high ranking individuals and asking whether that recapitulates some of the patterns that we observe. Good question.
On the baboons, we're just doing observational studies, or we're also doing DNA study on the baboon population?
So the paper that I talked about primarily today is based entirely on observational, demographic, and behavioral data. However, we do actually quite a lot of genomic work on the baboons, and I just didn't talk about any of that work today, and we haven't connected it directly with some of those results, although we're trying. Is there something in particular that you want to know about, or just... So, yes, yeah. So we have been collecting those sort of paired control and LPS stimulation type samples from animals in Ambacelli for the past 3 or 4 years. So collecting from the wild baboons is slow because we prioritize habituation, and so when we dart them, we have to dart them when nobody else is looking, and we maybe collect 20 samples a year or something like that. But we're gradually aggregating a sample set where we'll be able to look at some parallel questions, I think, in both species and ask in a completely natural environment whether you also can observe social environmental effects on immune responses.
For the LPS data, have you thought about looking at double-stranded RNA, which will also signal through TRIF, but may mimic virus infection better than LPS?
Yes, well, LPS mimics viral infection remarkably badly, and so one question we got was, but you're saying that you've got natural killer cells, and then you did an LPS treatment, right? Well, we didn't know those were going to be our results when we started. We started with LPS because differences in inflammation have long been hypothesized to be linked to social status. We have a gardiquimod infection using the same design, which I think is what, TLR7 and TLR8. I really wish we had done poly-IC, which we didn't. But I would do if we started it over again. So one of the things that we're talking about doing is expanding to a larger set of both ligands, like poly-AC, but also using heat-killed bacteria itself or potentially live pathogenic bacteria, which gets a little bit closer to in vivo, right? Basically, we're doing it this way because we're trying to stay as close to in vivo as possible without injecting our animals with stuff. The other thing that we've talked about doing, because these kind of studies happen at the primate centers all the time, is vaccination response, right? So, Yep, right, exactly. So this might predict something about variation in response to vaccines against virus. Yeah.
When you have a high status, and I'm thinking more of humans than people, so you're born into a high status, you eat better, you go to the doctor, you go to the dentist, is there almost like a circular loop of advantage causing DNA changes?
Right. So here I'm not talking about any change in the DNA. I'm talking about changes in how the DNA is translated into all the things that our proteins do, right? Or, at the level of gene expression anyway. So let me see. I think that, I don't study humans, but I think probably, there probably are cyclical advantages that are transgenerationally expressed that don't even need to invoke any mechanisms at a cellular level. There may be mechanisms at a cellular level that are involved, but it's clear that socioeconomically privileged people in the United States pay a lot for their kids to go to really expensive schools, and that's a source of advantage that predicts their kids' income, that predicts their kids' health care. And so certainly I think that advantage is propagated. Whether it's propagated with the sorts of mechanisms I'm talking about, I don't know. I mean, in fact, our switches in social status suggest that what's going on in your cells has to do a lot with what is happening to you right now, as opposed to what happened to you in your sort of social history. So that piece of data in and of itself would suggest maybe the mechanisms that we happen to be looking at, and these are only one type of readout of gene regulation, are maybe not going to be so stable over time if the environment changes. But I think that the question you're asking is an open and interesting one that many people would like to get into.
So the differences in plasticity you see in responses to adverse effects between early life and in adulthood are kind of interesting. And I'm wondering whether you think this is a species difference or whether there's something like an epigenetic modification that occurs in early life in a specific developmental window that closes before adulthood?
So there's a long history of work on early life effects, both in animals, non-human animals, and in humans, that suggests that we do have particular windows in development where we're particularly sensitive to insults. And so it, in that sort of very qualitative sense, maybe makes sense to me that early life environments seem to have such long-lasting effects, and that differences in social status switched over the course of just a year or so might not. But to really answer that question, we'd have to understand more about, one, whether the the plasticity and the effects of social status that I showed you are, for example, tissue specific. Turnover of cells in the immune system is much faster than turnover of cells in other parts of our body, although some immune cell types live for a long time. And so maybe you erase signatures of past history faster than you would, for example, in the brain. So we don't know whether this is a tissue-specific phenomenon, one. Okay, so I don't think we really know enough about epigenetic signatures of early life adversity I think the jury's still out on how important those are. Most of that work has been on DNA methylation. Much of it has been on humans, where there are lots of confounding factors. There are associated environments between early life and adulthood. So is it really just early life? I don't think we know. And this is like sort of a thing that bugs me a lot. We measure DNA methylation, and we have no idea whether it does anything else. So there can be lots of epigenetic changes, but those epigenetic changes arguably don't matter at all if they don't have a downstream effect on phenotype. And so we often don't know whether the regions of the genome that are sensitive, or they look like they're sensitive to early life environments, also have even an effect on gene expression, which would be the next step in the phenotypic chain. So, yes?
Although in rats, early life differences in parenting have an effect on stress response.
Yeah. So I'm actually not questioning that there aren't early life effects. I think I was trying to answer the question, is this epigenetically mediated? Right? And so the presence of early life effects, there's tons of evidence for the presence of early life effects.
Right, but that appears to be epigenetically mediated.
Maybe. Yeah. Okay. We're trying to ask those questions. So we'll see whether we find anything that I think is convincing.
Hi, yeah, I was wondering if the higher ranking rhesus monkeys get more food or higher quality food? Is there a difference?
So they are fed ad lib on delicious, nutritious primate chow, which is actually incredibly healthy for you, but I suspect tastes not very exciting. So we were curious about that too. So one thing we did was very carefully track their weight, every week of the study. And there are no differences in weight gain or weight loss between high-ranking and low-ranking individuals, suggesting that what we're observing is not at least a direct consequence of cumulative energy balance. That's not quite answering your question. So they're certainly not getting different types of food or higher or lower quality food. They're all eating the same food. But whether they have more uninterrupted access to food, that's possible because we aren't, some people actually put RFID tags in so they can track who's pulling the food. We don't have that sort of data here. So all I can tell you is nobody changed weight in a systematic fashion. So I think that's not what's happening. And also when we basically control for body mass by controlling for age, they're strongly correlated in our sample. Yeah. Thanks.